Thank you for participating in this study challenge. Here's a question from the USABO Semifinal exam June 24, 2020. Please drop a like, comment, share this out, and subscribe as we continue to do USABO together. The lysine side chain is charged and basic at physiological pH. Its pKa is around 10, so it is protonated in water. The hydrophobic pocket represents a difference in polarity. The pKa shifts to favor the neutral state, so that lysine can be a proton acceptor. Hence lysine pKa will be lower in the hydrophobic pocket. For acids like aspartic acid, the pKa increases in the hydrophobic microenvironment. This makes aspartic acid protonated, and allows it to function as a proton donor in the hydrophobic pocket. Thank you for participating in this study challenge.